You've seen our research on social media. Now, join us as we dive deeper into the public health topics of our time, featuring new studies and findings generated by our faculty and researchers committed to advancing health equity. From the Department of Population and Public Health Sciences at Keck School of Medicine of USC, this is Preventive Pros, the podcast.
So I'm Olivier Pernet. I'm assistant professor in the pediatric department, and I'm also the president of a nonprofit organization called Enviro International Laboratories that focus on virus surveillance and outbreak preparedness. And I have been working, recently mostly on SARS coronavirus 2, of course, and at USC mostly on how the virus can transmit from the mother to the baby during pregnancy, and also developing new assay that allows surveillance for new variants. So there was a real gap in the knowledge on how these viruses transmit because their coronaviruses are usually associated with respiratory disease. So we don't really look at them from mouth-to-tail transmission. But we knew in some cases the baby were born with already infected. So there was obviously transfer. So that's why it prompted us to look at that. And in the department, there's a long history that date from 30 years ago, focusing on how viruses transmitted during pregnancy. So it was a natural thing to do to study vertical transmission of SARS coronavirus 2. It's a little early to go into detail, but what we observed, first we had to develop correct assay because right now there's a lack of assay for neonatal diagnostics. So we have to develop new assays to refocus on the baby, the newborn. And what we're seeing with this new tool is that it's actually more prevalent than what we saw. There is results already published that varies a lot depending on the study, depending on the protocol they use, depending on methods. And being able to normalize all that, to standardize all that, start to yield some interesting results that the virus actually transmit more often than what we suspect. So we're really trying to see how that's going to impact the life of this newborn down the line by looking on long-term outcome. Is being exposed to coronavirus in utero has long-term outcomes. So that's one of the first things we want to look at. The second is there any way to prevent that? For example, we are looking at vaccine recall from this mother that consented for that study and see what combination of vaccine is helping preventing the virus from transmitting from the mother to the baby. So we are analyzing as much newborn cord blood as possible, either because the mother consented for the study, which usually is not really a problem. They're very happy to help us for that. But sometimes we just cannot reach this mother, but we have access to blood bank, and that's how we get access to that. So a rise of coronavirus cases is probably going to have an impact on what we see because we know some there are breakthrough cases, so we probably see that in our study. So we are focusing on emerging disease, and it's not only SARS coronavirus 2. There's a lot of other viruses that emerge. Actually, it's suspected that these emerging diseases are going to get more and more common. And what's very interesting, these emerging diseases are very often zoonotic disease, which means they originate from animals. So famous classic zoonotic disease would be rabies. So they get bitten by fox or dogs, usually it's a dog, and get infected. So it's transmitted from the animal to the human. Another famous zoonotic disease would be influenza, which translates from wild bird to usually farm bird, and then to human, sometimes with other intermediate. But it's really an animal disease that spread to human population and then to the rest of the humans. So different aspect of this. Zoonotic doesn't necessarily mean it's always from the animal you get infected. It can originate from an animal and then spread from human to human. So that's what we are studying. So there's definitely a rise. We estimate now that more than half of the disease in human actually originate from animal. The question is whether is it new or not. We definitely see an increase because of different changes in ecosystem. That's something we can come back later. And as you mentioned, also because now we have the ability to test and discover more of disease somewhere around for a long time. It's just we never really pay attention to it, or we never really detected it. There's also another factor is that now we're testing everywhere and hopefully as much people as possible. Just to give two examples. The first one will be HIV. When you think about HIV, we usually think about HIV starting late 70s, early 80s. It's actually a disease that was around since the early 20th century, but it was mostly located in remote part of Central and East Africa. And there was no really testing or so there was no technology and definitely no interest by colonial authority to look at that. So it's only when it reached the US and there started to be some US patients that HIV appear, but it was there from actually present for much longer. The second example would be the hantavirus that we have here in the US. So the Cynombre virus. Usually said it emerged in the early 90s, which is when it was discovered, 1993. But actually, we found records from Native American who have documented that from a long time ago. So there's also that issue that something had already been reported, just we start to pay attention much later. All right, so about misconception now. When we talk about emerging zoonotic disease, we think about terrible things like Ebola or Nipah, or even SARS coronavirus 2, of course. And they all have something in common. They all come from bats. So there's kind of a idea that bats are the sources of all our problem and regarding emerging disease. That's a misconception. It's actually the other way around. At the ecosystem level, bats act as a regulator, like a jail for viruses. They're unique for two reasons: for their genetic diversity. I mean, the second group in mammal diversity after rodent. And they're also very interesting for the immune system. The immune system is one of the most incredible immune system you can find in mammal. That's by far surpass everything and different from every aspect compared to other mammals. So these two things, these two parameters make them a perfect kind of guardian of the virome. There's kind of a misconception about virology in general, that each species has its own viruses, and then zoonotic viruses are kind of the exception. It's actually not the case. From a virus point of view, it doesn't really matter if you're a human, a gorilla, or a bat, or a horse. Or what's important is, can the cells be infected? So because now, because of human activity, we have connected all the ecosystem into one giant human ecosystem. Viruses have access to all the elements of that ecosystem at the individual level, population level, even at the cellular or molecular level. So the only parameter that matters is, can the virus interact with this cell? Can the virus interact with this protein? Can the virus interact with this population? So in that regard, now when you look at that, bats being kind of a jail for virus, if virus manage to get out of the jail, it can spread very quickly in the rest of the population because everything now is collected. So why would a bat release virus? Bats don't release virus. Bats protect us against virus, but the thing is, we don't protect bats. So bats' immune system decrease in efficiency, and that amazing immune system that was basically protecting us against all these viruses is now weakened, which means the virus can spread, the virus can move from one host to another, and we don't have that tremendous immune system. Other animals that are brought in contact with wildlife, especially bats, might be exposed to, and these farm animals are exposed to humans. So we have created a perfect chain of transmission from wildlife source, the bat, to human population. So all these ecosystem change that weaken bats' immune system are actually ultimately affect us. There is a very nice analogy that say that these outbreaks, recent outbreaks of bat-borne virus, are kind of the shingles of the ecosystem. Shingles is the chickenpox you get as a kid, and then when you grow older, your immune system decreases, and the chickenpox that was always in there waiting that your immune system decrease after decades finally has this opportunity and reactivate. It's kind of the same thing that's happening at the ecosystem level with this virus. They have been prisoner of the bat immune system for centuries, and now this immune system that kept them on check is decreasing. So now they can spread again. So that's a very nice, I think, analogy to look at this issue. So there's 3 steps here. First is at the bat level. So here is protection of the bat, protection of the ecosystem, just to make sure our ecosystem maintain their healthy bat population. That's extremely important. Second aspect here is the transmission between bats to wildlife to farm animal. In this case, it's really how to manage land and how you manage agriculture. That's going to be important to reduce the risk. And then there's at the population, human population level, by surveillance, predicting what's the next, what viruses are very likely to emerge, because we know for looking at viruses that are already circulating in wildlife or in domesticated animal, we can kind of predict which one are the chance to jump to the human population. So for the prevention, that's really all these 3 steps. What's very interesting is that's not new. It's not just me having a bright new idea that's going to save the world. We actually already do that for flu. Flu is a really, really interesting example for zoonotic disease. And we have taken flu very seriously. Flu, just in the US, I'll give you some number, kills 10 to 50,000 people a year. It costs US above 10 billions in just medical cost every year. That's not even considering the agriculture loss, because this virus also going to affect poultry in farm. Zoonotic diseases are estimated to cost half of the loss of the agricultural sector for animal agriculture. So it's a massive economical and human and public health cost. So for a very long time, influenza has been taken seriously, and we have incredible surveillance system at the wildlife level, at the domesticated animal, and at the population level. We have incredible surveillance system at the local level, at the national or regional level, and also at the international level by different collaboration. It's not very surprising that most of the tools that were the most useful during the SARS coronavirus 2 early stage of the pandemic are the one that were put in place for influenza surveillance. So we know how to do it. We're doing it for influenza. Now the goal is to really being able to do that for not every virus, but for having a more general approach and not just focusing on influenza, but all these viruses and also parasites and bacteria that can spread from animals to humans. So if you wait that somebody show up with a disease that it's already too late. I mean, you can, even if you have a very good, very strong response, unless you have a very strong response immediately, it's already too late if somebody show up with symptoms. The idea is really to prevent that, to monitor that in a month, upstream of the transmission chain. So here's again, surveillance is not at the human population, but to protect human population, you have to do surveillance at the environmental where this population lives. It's going to be very different if you do that in California, or if you do that in Malaysia, or if you do that in Cameroon. It's going to be very different. Another thing is the resources available are going to be very different. We saw that during the COVID-19 surveillance international effort. It was very different, the data you would get from the US, from the UK. Just two countries from the north, which we would expect to give similar results. There was very big differences. Now, if you look at even more a different country, like during a meeting, I was talking to somebody doing surveillance in Guatemala. The challenge that they are facing was very different from what we were facing here in the US. Not everybody have access to the technology that we have. And it's not, even if we have access to the technology, sometimes it might be more expensive to do that than other approach. So that's why this issue has to be addressed, taking into account the local context, taking into account what's possible to do. In Los Angeles County, we had a great sequencing capacity, so we could monitor what was circulating, which variant, which mutation. This was great. I mean, there's some amazing research being done. This colleague in Guatemala, they could barely get enough reagent to run their assay. So the idea is not to have a massive investment so everybody can do sequencing. That's not going to happen. I mean, it would be nice, but that's not going to happen. So it's very important to develop new tools that can be used everywhere. Like, for example, one of the things we've done at USC in the lab was to develop an assay that require a regular PCR, qPCR equipment, which almost everybody has. Like every hospital has such equipment, to differentiate between different variants very quickly. So that means instead of relying on a sequencing center that is very costly and rare, now every hospital with a minimum equipment could identify very quickly their variant. That's how you increase surveillance. You give the possibility, you give the protocol, the methods for identifying this variant everywhere. Now, this has to be applied to surveillance in general. Is there any technology that we can use everywhere in the world to identify what's circulating? So that's one of the challenge. For example, I've been working with a group in Gabon doing Ebola surveillance. They have to go collect gorilla samples. So pretty far from any advanced technology. I mean, gorilla don't live in university campus. They live deep into the forest. So these collaborators, they have to adapt their protocol. They have to adapt their methods to what they're doing, to their local environment. And I think that's very important, that we do not focus on what would be the best technology, but what would be the most ubiquitous technology. And then finally, having all the results from all these different technology being able to be compatible. So if I combine the data from a lab in Southern California, it will be compatible with Northern California, but also be compatible with what's been done in Northern Mexico, for example. These emerging zoonotic disease, they are not new. Actually, it started as soon as humans start to interact intensely with their environment. At some point in Sumer, 4,000 or 5,000 years ago, people thought it was a good idea to put a lot of humans together to create cities. And that's actually a good idea that happened at different places around the world. But what's unique about Sumer is that to maintain this urbanization, this new thing that was urbanization, they had to also maintain a different kind of agriculture that can feed everybody in this city. So they start to have bigger farm with more cattle, with more animals. And because now they had a concentration of cattle, there was more virus circulating. And one of these viruses circulating somewhere is mutated and was able to jump into human. Now, 5,000 years ago, we still have to deal with it. It's called measles, and it kills 100 to 200,000 people a year and leave a lot of other with blindness, deafness, and immune amnesia. So this is not new, and it's not like there's an outbreak and 4 or 5 years later it's going to be over. Some of them are going to stay for a long time, and we still have to deal with it, and we're still going to have 100,000 of deaths every year because of this disease. So it's a serious problem. It's not going to get better, even if we change it. Everything we're doing wrong right now is going to take years and decades to reverse. Meanwhile, bats don't have the time to evolve that fast. It's not only the bats; a lot of other animals, the ones that are very close to, like apes, also have their own virus that can no shade. Rodents also. Rodents, not just rats in Los Angeles. There are also a lot of different rodents. That's the most diverse group. So they have their own virus too. And that's not surprised that bats and rodents are usually associated with disease. It's just more prevalent than any other animal. So that's why, and we are directly exposed to rodent. There is some extremely dangerous virus circulating in the hills just behind us in Los Angeles. So it's not something that we should think of. It's a problem in Southeast Asia because flu, because COVID. It's a problem in Africa because of Ebola. It's a problem of India and Bangladesh because of Nipah. No, it's something that's everywhere. Everywhere ecosystems are damaged. Everywhere animal population under stress. Everywhere there are new interaction between wildlife and domesticated animal and human. It's a problem. So we really need to have this One Health approach when we look at all the scale, local, regional, and global, for everybody involved, which means people working on wildlife, people working with domesticated animals, and people working with human, in order to achieve the best public health policy. It's a really international and multi-level work.
Preventive Pros, the podcast, is produced by the Department of Population and Public Health Sciences at Keck School of Medicine of USC. To learn more about any of our episodes, or to subscribe to our monthly Preventive Dose newsletter featuring the latest in public health research and news, visit pphs.usc.edu/podcast. Thank you for listening.